Hello guys, my name is Dr. Abid and today we will study a lecture which is called Basic Definitions in the Immunology section. So let's start. What is immunology? Immunology is the study of protection of our body from the macromolecule and invading organism and our response to them. So what it's mean that for example this is a bacteria and that enter into our body. Let's say that this container is our body. So this bacteria enter our body and our body have a response against that bacteria, that a particular response we will study in the next slides. So our body have a response against that bacteria and our body will kill that bacteria. Not only our body kill that bacteria, but its particle also cleared by our body. So that ability of our body to protect against that bacteria is called immunology. And not only in immunology we study the only ability, but how our body make a response against that bacteria. We will going to study in immunology how innate immunity and adaptive immunity, or innate immune response or adaptive immune response work and clear the macromolecule and invading organism. So next definition is immunity. So what is immunity? Immunity just say that the ability of our body to protect the diseases. It is just ability of our body to protect against the diseases. That is what we call immunity. Now immunity have a lot of types, but here we will discuss that only two important type of immunity. One is called autoimmunity and other is called tumor immunity. In autoimmunity, immune response against our own protein will develop, and that what we are calling that autoimmunity. But in tumor immunity, our aberrant cell, or we can say that tumor cells, if our immunity develop against the tumor cells, that what we are calling that tumor immunity. So next definition is what is immunogen. Immunogen are those molecules which produce immunity or immune response. For example, different type of antibody which are produced by our body, they are also immunogen. So are different antigen, they also can be immunogen. So antigen may be immunogen or may not be. For example, heptan molecule which don't have ability to produce immunity, so they are not immunogen but can react with the specific antibodies. So next definition is antigen. So what is antigen? This word we will study in a lot of time in immunology. So we have to clear. So what is antigen? So and antibody. So what is the difference between two of them? So first definition is what is antigen. Antigen is a toxic or foreign substance that induce immune response in the body. So that is what called antigen. So antigen is a foreign substance and can be a toxic substance that produce an immune response in the body. That is what we are calling antigen. I said that one bacteria, I just give in previous slide an example that one bacteria that enter into our body, and that bacterial wall have a different protein that could be a antigen. Our body can make response against that bacterial surface or bacterial membrane. So that what we are calling antigen, or maybe not full bacteria, maybe that bacteria was broken down into pieces by our body, and few fragment of that bacteria, for example these are the fragments. So that fragment could be a antigen, or any toxic substance which enter into our body, that could be a antigen. So antigen produce immune response. Antigen produce an immune response. And what is that immune response? It could be a production of antibodies when antigen or toxic substance attack in our body, and our body have a response that they will produce antibodies. So what is antibodies? The substance produced by the cell to kill the antigen. So our body make a lot of antibodies against the antigen, and that will neutralize that antigen. And how are we gonna show that antibody showing can be shown like a Y shape. I am making this like a Y shape, and this is our antibody, it's shown like this. And they help in neutralizing the antigen and neutralizing a lot of bacteria. We have a separate full lecture on the antibodies, so we don't have to worry that we will have a whole lecture about antibody, how they develop, and how they help in clear the bacteria. So we will have a full lecture on antibodies, so we don't have to worry of that. So if we go to the next slide, so we said that one bacteria, we are saying that, for example, this is a bacteria that is enter into our body. For example, this is a container that enter into our body. So this bacteria into our body, and our body have a response against that bacteria. So immune response can be two type. One is the innate immune response, and second is the adaptive immune response. Or you can say that innate immunity or the adaptive immunity. So let's discuss one by one. So what is innate immunity or innate immune response? Again, we will take an example of a bacteria. For example, this is a bacteria, and this bacteria into our body, for example, like this bacteria into our body have a first response against that bacteria. So our first response will be the innate immune response or innate immunity. How will be the first response? That macrophage, neutrophils, macrophage, monocyte, dendritic cells, these cells naturally produce in our body. We have studied in hematology section that how these cells are produced in hematopoietic lecture. So these cell will immediately kill that bacteria. So that response is, we are saying that response is non-specific, and that is immediate response occur into minutes and hours, and that don't have any memory. So we will discuss that how they don't have a memory when we will study and compare innate immunity with adaptive immunity. So what is innate immunity? We have a just definition, got that definition, that is a non-specific and occur rapidly in minute to hour and don't have any memory. This is just a simple definition of innate immunity. And what are the players of innate immunity? What are the cells of innate immunity? What are the component of innate immunity? So these component we have to remember that is a first component is first cell is neutrophil, then macrophage, then monocyte, then dendritic cells, and then natural killer cells, and complement and physical epithelial barrier, and secreted enzymes. So these are the component of innate immunity. So how actually they kill this bacteria, these cells, how they gonna kill that bacteria. Actually these cells have a receptor on his surface. For example, this is a macrophage have a receptor, specific receptor, that is what we are calling toll like receptor, TLR, T for toll like receptor. So they this macrophage have a toll like receptor, and this receptor will gonna bind with that bacteria. Just we give an example before that, which bacteria enter into our body. So this bacterial surface will attach with this toll like receptor. For example, this bacteria is a gram negative bacteria. For example, this is a gram negative bacteria, and gram negative bacteria have a lipopolysaccharide, special lipid on the gram negative bacteria. We will study in the microbiology section, so no, you don't have to worry. So lipopolysaccharide, we call that toll like receptor bind with the lipopolysaccharide of the gram negative bacteria and produce an immune response. Maybe this macrophage can engulf this bacteria, phagocytose this bacteria and kill that bacteria. So this is a very clear mechanism that how our body will in innate immunity will kill that bacteria. So this lipopolysaccharide or maybe some other component, they also called PAM. PAM mean pathogen associated molecular pattern. So what is happening there, this toll like receptor of a macrophage can be recognize the bacterial surface PAM molecules. In our example, in particular our example, we said that gram negative bacteria have a lipopolysaccharide. So lipopolysaccharide will attach with the TLR and kill that bacteria. So what is toll like receptor? That pattern recognition receptor that recognize pathogen associated molecular patterns. So this TLR will bind with PAM. I already give in this example that this TLR of a macrophage can bind with PAM, and that lead to activation of NFKB, a substance which active and that help in clear that bacteria. What is the example of PAMs? PAMs could be a lipopolysaccharide in the gram negative bacteria. I give already example, that example I already have given. And could be a flagellin in the bacteria, or a nucleic acid in the virus. We know that the viral have a DNA. In DNA there could be a RNA or could be a DNA. They made up a nucleic acid. So these virus nucleic acid and lipopolysaccharide and flagellin, they example of PAM. So I'm gonna repeat again that section, that for example this is macrophage, and macrophage have a toll like receptor, TLR receptor, and that receptor will bind with that particular bacteria. For example this black bacteria. So the black bacteria have a LPS, lipopolysaccharide. This lipopolysaccharide that we are calling that in gram negative bacteria, these are the PAM receptor. So TLR will bind with PAM receptor and produce a response, maybe activation of NFKB. I can write here NFKB. So NFKB, so the activation of and kill that bacteria. And other component which is produced by these our player of innate immunity, our cell of the innate immunity, maybe lysosome. Lot of macrophages, produce lysosome. Neutrophil also produce lot of lysosome, and other are the complement system and C-reactive protein and defensin, which is a protein which is produced by the natural killer cells, and they also help in the clearance of bacteria. We will, in the lymphocyte lecture, we will study that how now natural killer cell work and kill the bacteria. Just trust me that now that defensin, which is proteins which is produced by the natural killer cell and kill that bacteria. And the next bullet is that this innate immunity is a germline encoded. What is meant by germline encoded? That our body have a natural ability to produce these layer of innate immunity, these cells of the innate immunity, and these cell help to clear the bacteria, virus, and any pathogen which enter into our body. So that is our in part of DNA, that we can say that help in hematopoiesis and produce these kind of cells, and these cells will help in the innate immunity to clear these bacteria. So that what it's mean by germline encoded. And once, for example, sometime it's happened that this bacteria, the black bacteria, will produce a resistance that against particular cells or a drug which we are take by the pharmacist. So these bacteria can be resistant against that particular cells or that particular human being. So our immune system will not work and produce this bacteria will produce a resistance, and that resistance will persist throughout the generations and does not change with the organism lifetime. So that organism lifetime does not change, and maybe these bacteria will produce a capsule around themselves. So they produce a resistance against our body, or we can say that against the drugs. We have a example. For example, one is the MRSA, special kind of bacterium. MRSA means methicillin resistant staphylococcus aureus. That is a staphylococcus round bacteria. The staphylococcus aureus have a resistance against a drug which is called methicillin. We will study in the pharmacology, you don't have to worry that. So these resistance will persist throughout the generation. So all the human being have a methicillin resistance bacteria have a resistance against the all human being, and when we are give to the that such patient methicillin. So that's clear that resistance persists through the generation and does not change with the organism lifetime. So that's it. That's all the thing which we have to know about the innate immunity. So let's compare the innate immunity with the adaptive immunity. So what is adaptive immunity? Adaptive immunity is a highly specific, refined over time, and develop over the long period. So what it's mean that for example, this is a bacteria which enter into our body. For example, this is our body. This bacteria enter into our body. First response will be the innate immune response. After the innate immune response, if few components are not removed by the innate immunity, so adaptive immunity will come into play, or they work as a side by side. Adaptive immune response or innate immune response will work side by side. So lot of antigen, bacterial antigen will be produced So for example, one antigen is round, this is a star antigen. So lot of antigen will be produced. I am showing here too. So against these antigens, our body have a response, that maybe T cell will differentiate and colonal expansion will occur of the T cell. Lot of T cell, just it's mean that lot of T cell will be the come into play. So lot of T cell will produce, and these T cell will help this antigen to clear, and maybe some plasma cell come into play also, and plasma cell will secrete antibody. Actually the B cell which is developed and mature, that is called plasma cell, and plasma cell will secrete antibodies. Antibody will bind with these bacterial antigen and neutralize them. So that's a long process, that production of antibody. We know that antibody are peptide chain or protein. So in the production of protein, that will take a long time, and in the clonal expansion or these colony of T cell, particular T cell, these particular T cell will neutralize that antigens, and that have a long time. So these T cell take a long time to expand its progeny, and remove that bacterias. We know that in production of lot of T cell and proliferation, it will take a long time, that DNA will synthesize peptide chain of a particular membrane are synthesized, and cell make its size bigger. We will study in cell division in the histology, so how cell will divide. So this expansion, clonal expansion will occur, and it take a long time, so it develop in long time. So next time this type of bacteria will enter into our body, so we have a pre-synthesized antibodies. Pre-synthesized antibodies, we already have a pre-synthesized, these are the pre-synthesized antibodies, and we have selected clonal expansion of a T cell to kill this bacteria. So that response will be the robust response and a faster response in adaptive immunity. So that's what we are saying in the definition, that this is a highly specific and refined over the time, and have a more faster and robust response produced by the memory cells. So what are the memory cells? Memory cell are just activated B and T cells, which is exposure to that antigen or that bacteria. So what are the component of the adaptive immunity? Adaptive immunity, we already have discussed that is a T cell, B cell, and circulating immunoglobulins or antibodies. So these B cell and T cells against this bacteria, clonal proliferation, we said that clonal proliferation will occur. So this clone are specified for that bacteria, and they have a receptor on the surface which is produced by the VDJ recombinations. So these receptor will attach with that bacteria and help to kill this bacteria. So VDJ recombination happen during the lymphocyte development. What is VDJ recombination? V is for variable, and D is for diversity, and J is for joining. So for example, this is a lymphocyte, and this lymphocyte have a gene which is called VDJ, variable diversity and joining region. For example, this is the V, this is the D, and that third is the J. I make here red. So VDJ, variable region, and diversity region, and joining region. So these region are like a shuffling, like let's say they have a different combination, like we play a card and we make a different combination with card and play. So this gene is also playing like this. They make a different nucleotide recombination, and when for this nucleotide is translate into messenger RNA, and messenger RNA will translate into protein. So this protein, specific protein, will inject into the surface of lymphocyte, and they make a receptor. So different receptor will produce by these clonal expansion of a T cell. So this different receptor produce against that particular organism or bacteria. For example, this clonal expansion have a we say that star receptor. Some have a V-shape receptor. Some have a cup shape receptors against that bacterial surface antigen, and a different response will be produced by these expansion or these progeny of a lymphocyte. So what is the benefit of VDJ recombination? So benefit of VDJ recombination is that every time when a bacterial antigen enter into our body have a make a different unique type of response against this bacteria. So for example, I make a different response against this bacteria, and some other person have a different response against the same antigen which enter into our body. So this due to this these are like if bacteria have a resistance against that colony, that will not persistent throughout the generation of a human, because every time this recombination is produced and person have a different response against that particular bacteria, so resistance will not be persistent throughout the generation, because a unique type of receptor is producing by these lymphocyte colony, and my response will be the different against that bacteria, and other person have a different response against that bacteria. So we already covered that bullet, that microbial resistance is not heritable. So for example, one bacteria is resistance with this specific lymphocyte colony, maybe next person make a different colony of a lymphocytes that will have a different response and kill that bacteria. So microbial resistance is not heritable. We already covered that memory cell, which is activated B and T cells. What they produce a memory which help in adaptive immunity, that are enter into our body, and that bacteria will be killed and by the robust response and pre-planned response, which we are calling that memory response, that produced by the activated B and T cell. So that response will be the more robust and quicker immune response will be produced by the adaptive immunity. Thank you very much, guys. So this is a new year gift for you. Thank you for watching my lecture.